Assalamu alaikum students, I'm Fauzia Iqbal, your biology teacher from Multan Public School and College for Boys and Girls. Dear students, this is your chapter number 5 and topic is cell cycle. We have gone through first phase of mitosis, that was karyokinesis, and today we will discuss about cytokinesis. We will start from this topic. This is cytoskeleton. This is the division of cytoplasm. It takes place differently in animal cell and plant cell, as here you can see that in this diagram also. In animal cell you can see that cytokinesis occur by a process known as cleavage, and a cleavage furrow develops where the metaphase plate used to be, means where the metaphase plate was formed. There a deep tube like structure appears which is known as furrow, and it is going to pinch the parent cell into two daughter cell. Pinch mean it is going to divide the parent cell into the daughter cell. And in plant cell you can see it is different from that of animal cell. Here you can see that there are some vesicles which are being produced from the Golgi apparatus. Those move to the middle of the cell and fuse to form membrane folded disc, and which is called cell plate, and it is also known as phragmoplast. The plate grows outward and more vesicles fuse with it. Finally, membranes of cell plate fuse with the plasma membrane and its contents join the parental cell wall. It results into two daughter cell, and each daughter cell is bonded by its own plasma membrane and cell wall. Here you can see the difference in both plant cell and animal cell, that animal cell is going to separate from each other but plant cell would remain with each other. Now the next topic is significance of mitosis. Here you are told that importance of mitosis is the maintenance of chromosomal set, means chromosomes should remain alike in composition and equal in number to the chromosomes which are present in the parent cell. And here different occasions are discussed about the lives of different organisms where mitosis is taking place. First point is development and growth. Here in this diagram you can see that there is a single cell which is called zygote, and it is going to divide by mitosis, and repeated mitosis would take place and it would convert into a ball of cells, and from ball of cell it would differentiate into new developing baby. And after the birth of this newborn baby, it would increase in size again by mitosis. So development and growth, both processes needs mitosis. Now this is the second point of significance of mitosis, that is cell replacement. In some parts of our body, for example our skin and digestive tract, cells are constantly sloughed off and replaced by new ones. Sloughed off mean broken down, and new cells are formed by mitosis, and so these are the exact copies of cell being replaced. Similarly, in our body, red blood cells are formed for very short lifespan. For example, it is of 4 months, and after 4 months new red blood cells are formed by mitosis. So these are the examples of cell replacement and from our body, and here mitosis is taking place. Next point is regeneration. You can see this regeneration in this diagram also. Here you are shown planaria and hydra and starfish, or which is also known as sea star. In all these organisms, the lost body parts are replaced by mitosis. They have this capability that they can make their lost body parts by mitosis. Next and last point of significance is asexual reproduction. Asexual reproduction takes place, for example budding, vegetative propagation. All these processes are being happening by mitosis. As asexual reproduction, one example is given you as vegetative propagation. Vegetative propagation takes place in plants. These plants increase their number from their body parts. As you know, we use the stems, roots and leaves of different plants to make new plants. This is known as vegetative propagation, and here you are shown grafting. In grafting, we are using the technique that two different plants are combined to make a new variety of plant, and here this process is going on by mitosis. Here you can see that this is again a vegetative propagation method. This is known as layering, that one branch is buried in the soil and it would give rise to the new plant. And the same technique is used for by stem cutting. And all these methods are called vegetative propagation for plants, and it takes place by mitosis. Here you can see asexual reproduction in animals. Here you are shown the example of HYDRA, that on one side of HYDRA an outgrowth is made. This outgrowth is known as bud. So HYDRA reproduce by budding, and this bud would increase in size and it would take the shape of HYDRA, and at last it would be detached. So these are the different examples of asexual reproduction which are taking place there by mitosis. Now I will tell you about errors in mitosis. Errors in mitosis is discussed for this purpose, that sometimes some sudden change takes place in genes. Actually, the genes are controlling all type of cell divisions, and when there is some sudden change in genetic material, this is known as mutation. Due to mutation, the cell continue to divide without any need. It results in the growth of abnormal cells, and it will become an abnormal mass over there, and this is known as tumor. You can see this in diagram. You can see the normal cells, and in the center there are some cells which are abnormally being produced, and this is known as tumor. In your book, you are told as long as these tumors remain in their original location, that is known as benign tumor. But if these invade other tissues, invade mean these are transferred to the other tissues, they are called malignant tumor. Malignant tumor is cancerous, and these cells are called cancer cells. Such tumors are can send cancer cells to other parts of the body where new tumors may also be formed, and this process is called metastasis. Metastasis means spreading of disease, which is very much dangerous. Tumor simply is not that much dangerous, but metastasis and malignant tumor is very much harmful, that is very much dangerous for health. Now our next topic is meiosis. Meiosis is other type of cell division which takes place in germ-line cells, and this takes place in our sex organs for the formation of gametes. This happens in plants and animals both. Here you are told that meiosis is the process by which one diploid cell, eukaryotic cell, divides into 4 haploid daughter cells. As here you are told that diploid eukaryotic cells means the diploid means having pair of homologous chromosomes are present in the form of pairs, and these pairs are called homologous chromosome. Here you can see 2 pairs of homologous chromosome. We will define homologous chromosome as those chromosomes which are similar to each other with respect to their size, shape and position of centromere. And here you are told that when some cell is diploid, it would be having these type of homologous chromosomes, but in case of haploid, only one of these 2 would be present in them. The word meiosis comes from the Greek word meions, which means making smaller, since it results into reduction in chromosome number. As a result of meiosis, number of chromosome becomes half in the daughter cell, so that's why it is called reduction division also. Now we will go for the phases of meiosis. You are told here that Oscar Hertwig, a German biologist, in 1876 first of all described it, and the preparatory steps of meiosis are identical to those of mitosis. Means there would be G1 phase, S phase, and G2 phase, and then interphase is followed by meiosis 1 and meiosis 2. Yes, meiosis takes place in two steps. First meiosis 1 takes place, and then meiosis 2 takes place. So first we will discuss meiosis 1, and in meiosis 1 we will again go for karyokinesis, and karyokinesis is divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. First of all, I will tell you about prophase 1 of meiosis 1. Prophase 1 is the longest phase in meiosis, and during this stage chromatin condenses into chromosomes. Homologous chromosomes line up with each other and form pairs by the process called synapses. Each pair of homologous chromosome is called bivalent, and each bivalent has 4 chromatids, as you can see in this diagram also, and this bivalent is also called tetrad. The two non-sister chromatids of homologous chromosome join each other at certain points along their length, and these points of attachment are called chiasmata. Okay, here you are to define synapses, bivalent, and chiasmata. Okay, these are the important definitions over here, and you can see their structures and the process in this diagram also. Now I will tell you about crossing over. Crossing over is the next step of prophase 1, where non-sister chromatids of homologous chromosome exchange their segments, and the phenomena is called crossing over. What is the advantage of this crossing over? Yes, it is very much advantageous. Here you can see in this diagram also that homologous chromatids are going to overlap each other, and they are going to exchange their parts, and you can see this phenomena by the change of the colors of the chromatids, and the exchange of segment result in recombination of genetic material. Genetic material is recombined, means exchange. It means it would result into change in the characteristics in the next generation. That's why crossing over is very much important phenomena in prophase 1. Next, you are told chromosomes condense further, and the nuclear envelope and the nucleoli disappears or regenerates, and centrioles duplicate. As that was duplicated at the interphase, those would migrate to the two poles of the cell, and kinetochore spindle fiber would be made, and chromosomes, and all chromosomes have kinetochore on them, so these chromosomes would arrange on the kinetochore spindle fiber, while the non-kinetochore spindle fibers from each side interact with each other, and two kinetochore spindle fiber from the opposite pole attach with the pair of chromosome. In mitosis, we have seen that two kinetochore spindle fibers attach with one chromosome. Okay, so this is the difference of attachment of chromosomes on the spindle. Here you can see the different phases of meiosis. At prophase 1, prophase 1 is completed. Now I will tell you about metaphase 1. The pair of homologous chromosome align along equatorial plane, forming the metaphase plate. Here you can see that homologous chromosomes are being arranged on the metaphase plate, but these are not in a single line. These are making in the form, these are going to align or arranged in the form of pairs. So this is the metaphase 1 of meiosis 1, which is different from that of metaphase of mitosis. Now anaphase 1, kinetochore spindle fiber shortens, and it results in pulling apart the chromosomes of each pair. Since one chromosome is pulled towards one pole, two haploid sets are formed. Each chromosome still contain a pair of sister chromatids, means these parallel setting of homologous chromosomes would separate from each other. Out of these two pairs, one would move towards one pole and other towards the other pole. Here you can again see metaphase 1. I have shown you here that two homologous chromosomes are arranged in the form of pairs but not in a single line. So this is different from that of metaphase of mitosis. You can see the anaphase also, that these two pairs of homologous chromosomes are getting separate from each other, but chromatid is not going centromere is not going to divide. Chromatids are not getting separate. Only pairs, out of these pairs of homologous chromosome, one pair is moving on one side and other pair is moving on the other side. Here you can again see the same process with the same setting, but you can see it that what is happening at telophase 1. Telophase 1 chromosomes arrive at the poles, and each pole now has half the number of chromosomes, but each chromosome still consists of two chromatids. Spindle network disappears and nuclear envelope is formed around each haploid set. Chromosomes uncoil again into chromatin, and cytokinesis now will take place, means pinching of cell membrane in animal cell or the formation of cell plate in case of plant cell would take place, and as a result two haploid daughter cell is formed. This is the karyokinesis of meiosis 1 completed, and then we will discuss about interphase. Interphase is also known as interkinesis. This is interphase 2, and interphase 2 is different from the interphase of mitosis and meiosis. In mitosis and meiosis, this interphase is different, because here in interphase 2 there is no S phase, because there is no duplication of chromosome required here. Now I will tell you meiosis 2. Meiosis 2 is the second part of meiosis, and it is similar to mitosis. It is divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. Prophase 2 takes very less time as compared to prophase 1. In this prophase, nucleoli and nuclear envelope disappears and chromatin condenses. Centrioles move to the polar regions and make spindle fibers. In metaphase, chromosomes attach with the kinetochore spindle fibers and align at the equator of the spindle. Again, you can see that they are going to arrange on a single line. This is followed by anaphase 2, where centromeres are cleaved and sister chromatids are pulled apart. Now the chromatids are getting separate and centromere is going to divide here. The sister chromatids are now called sister chromosomes, and they are pulled towards opposite poles, and telophase 2 is marked with uncoiling of chromosomes into chromatin, means again chromatin is made and nuclear envelope reform. Cleavage or cell wall formation eventually produces a total number of 4 daughter cells, and each with the haploid set of chromosome, means half number of chromosome with respect to that of parent cell. This is the end of meiosis, and now I will tell you some homework. You are given some definitions to write on your notebook, like phlegmoplast, regeneration, benign tumor, malignant tumor, metastasis, meiosis, synapses, crossing over, and metaphase of meiosis. Okay, and along with that you will draw crossing over and different stages or phases of meiosis on your notebook. This is the end of today's work, dear students. You can ask me any question in your meet class also. Thank you very much.